Okay, so welcome everybody. My name is Mark Gardner and we're going to kick off the meeting. So also welcome to Matt Todd from the University of Sydney, and it looks like Paul Willis may struggle to join us from his hotel, but I think we have the information covered. So I will give a very brief introduction. So hopefully the mechanics of webinar are reasonably clear to you. You will all be muted. If you want to speak, please click on the hand icon. It would also be fantastic if you can make contributions, questions, or other contributions, opinions through the questions panel. They do get recorded for us, so we can take a look at them afterwards and even perhaps follow up with clarification questions if need be. The agenda today, we're going to go through the brief project background and the scientific objectives, and if Paul can't join us, I think Matt will do that, and then lead us through the information sources. I'm going to show you a little bit of some analysis of some of the data with Data Warrior as one option, and then Matt's going to get into the bulk of the meeting, which will be an overview of the SAR and some 6, I think, key questions that Matt has about the project. This is something of an experiment. This is the first time that we've done this in what is now the 5th Global Health Compound Design meeting. It's the first time that we've really dived into a project and looked at some data and given you an opportunity to contribute your ideas about how the project is going, which we're really looking forward to getting your input to that. So that'll be the bulk of the meeting, and Matt will also give you some information at the end about submitting more ideas and input and what will happen to them. So there are lots of ways to provide input. The question box during the session, and Matt will also provide lots of information about how to provide thoughts and ideas afterwards through GitHub. So I will switch the meeting over to Matt. And Matt.
Great, thank you, Mark, and thank you everyone for showing up at whatever time of day it is for you. Really appreciate your time and potential inputs to the project. This is a live open source project, which means that in a nutshell, everything that is done is shared, so all data and ideas are shared. Anybody can participate to do anything. There won't be any patents. So these are the sort of 3 crucial rules about the open source project, about the way it works. So if you want to participate and you want to do anything with the data or make molecules or make suggestions, you can do that without asking permission. It really tries to adhere to open source principles that are well known in software. So in essence, it's a rather unusual project, and we are obviously trying to show that this works as a method of doing drug discovery and development. That's the sort of big picture part of the project, the way in which the science is done. Now, I'm very happy to talk about all of that stuff at length with anyone who wants to engage with it, but for this meeting, I think I'd really like to focus on the science and on several of the more pressing scientific questions that we have. And those were the ones that were shown on the deck that were sent around, and we'll work through these in turn. Just at the outset, I guess I'll say a few things about very generally about the project and what we're trying to achieve. So we are trying to do drug discovery and development for the treatment of malaria, small molecules for the treatment of malaria. We're obviously doing that in the public domain. We are starting with public domain starting points and improving those compounds so that ideally we could take a compound into clinical trials. That's the sort of aim of the project. We've been working for a few years now and have completed 3 series to varying degrees, and we're now working on the 4th, which is the subject of today's webinar, so-called Series 4, the triazole pyrazines. These compounds arose from a screen that was done of compounds that originated in Pfizer, and a very interesting set of compounds were identified. The data were passed to MMV a few years ago, who pursued the series a little bit with the resources of a contract research organization. That project then completed, and the data that were obtained by MMV with the contract research organization were passed to this open source malaria consortium about 2 or 3 years ago for us to pursue those as the main focus of our activities. Now, what we're doing then is using we have been using a government research grant here in Australia and resources from MMV to pursue that. So I guess the Sydney lab, where I am and where a few other people on this call are, is the one sort of grant-supported research lab, and we're trying to drive some of the synthetic chemistry and other things. But the aim of the open source aspect of this is that we're trying to generate this momentum at the heart of the project, but of course anyone else is allowed to contribute and do whatever they wish for the project and change the direction of the research if it's seen that it's not the most fruitful things we could be doing. So we're meant to be the sort of center of a much larger distributed set of people who are free to act as they wish. Now, I'll mention a couple of things about that maybe towards the end. The objectives of the science that we're doing, beyond all of the open source, how open source works, is to try and develop a late lead compound for this series. And this is something that Paul would have spoken about had he been with us. Now, what that means is the compounds that we want to synthesize and validate meet the criteria for late leads that are described on the Medicines for Malaria Venture website. And so the numbers are shown there, but in brief, the potency of the molecules that we're looking for needs to be under 100 nanomolar. Ideally, solubility is above 100 micromolar, and efficacy, ED90, is below 10 mg per kg. Short-term goals would be to get a better rodent half-life than we have at the moment. In other words, a half-life of more than 2.5 hours. So these are numbers that are well described on the MMP website. And combine some of these features obviously in one molecule. We can say, well, this is definitely our late lead, which we can pursue further. We are just shy of some of these things, as we'll talk about in this meeting. And though we have excellent data from the compounds in this series, we're not quite there yet. And obviously, in projects of this kind, as time passes, you can take comfort, of course, in the good data that you have, but at some point you have to sort of cross the line and get to a point where you can pursue the series further. So that's the main objective, is to progress the compound by improvements in potency, solubility, and reduced metabolic clearance. Those are sort of three things, and so those are the three things that I think we're going to focus in on today, more than other issues. So we can discuss some of these things in a moment, but what I'd like to do first, as the next thing on the agenda, is just to discuss some information resources, so discuss a little bit about how we can work together and how this project works. So where various bits of data are and what you can do with them, because that determines how we can communicate with each other. In open source malaria, over a number of years, we've been experimenting with a bunch of different things. We've never adopted a kind of monolithic collaboration platform because everyone wants different things. So we've taken a much more democratic, I suppose, view of how to work together. Adopting certain key things, but also making it clear that if you prefer to work with other tools, that's fine, provided everything is in the public domain. So the crucial thing, if you do any experimental work, for example, is that your laboratory notebook is online and is public. So every experiment can be seen and all the data are there. This solves trust issues because you don't need to trust anybody if you can see all the data that people have generated. So I'll just, hopefully the screen share is working here. I'll just shift to a couple of resource things. Hopefully, with the stuff that we distributed, you saw that much of the description of the meeting is on a platform called GitHub, which is something that's used by software people. We've sort of pirated the discussion forum on this thing. Normally, it's a platform that's used for sharing code and working on code together. But there's an issue tracker where people can raise things that need discussing or solving, and we're using that to discuss a lot of things on the project at the moment. We find it's a pretty useful platform for that. So a bunch of the descriptions of the different things we're talking about today are hosted on GitHub, and there is an issue list here of everything that's currently open, of stuff that needs either discussion or needs to be solved, and when things are solved, they're closed, and it works quite well as essentially as a public to-do list. You do have to have an account here, which is annoying, and there is at present no sort of daily digest of issues that have been raised, which can mean that you get quite a few emails. The laboratory notebooks, again, I've linked to a few of these on the materials for this meeting. One public lab book is this thing called Lab Trove, which we've used for many years, which is a pretty simple platform for sharing experimental work. It has issues to do with it. You can't search on chemical structure and so on. There is another that some people are using, which is this Lab Archives electronic lab notebook. So Chase Smith in the US is using this, and Ed in Sydney is using this too. A slightly fancier platform, again, not chemically searchable. But of course, if you want to do work on this project, you can use whatever lab book you like, provided that we can see the details. The data that's generated for the molecules in the project, so any synthetic intermediate is listed, should be listed on this Google sheet, which again is linked to in the preliminary materials we distributed. And all the biological activity that's associated with all molecules is also shown. It's a rather extensive spreadsheet because of that. But all of the associated data here are available, can be downloaded as an Excel sheet, and can be visualized with a link that we distributed in Luke Pettini's platform, ChemInfo. And this is a very useful addition to the project that Luke contributed, because his system allows a dynamic view of all the molecules in the project. So in other words, once you update the Google sheet, that's automatically reflected in the sheet that's here. So everything is immediately updated, and you can browse the molecules and download any particular series that we worked on. So here is series 4. You can download this as an SDF and manipulate the data as you wish. So that's been a tremendous addition to the project, to be able to visualize the molecules like that in a dynamic sense. I guess the two other things I wanted to highlight was that the summary of the series as it stands is on this wiki. So Open Source Malaria has a page for each of the series it's done, and this is the main one for Series 4. So this is a good place to sort of browse structures and what is known of the data that were inherited, and the new data as well. It can be good food for thought for what to do next. This is updated as much as we can. Sometimes we don't update it immediately. But of course, it's a platform that can be updated by anyone also, so this can be changed and added to and improved. And lastly, the landing page, which is the thing which brings people from Google searches, is just a page with the bare minimum of information on it, and which is auto-populated in many ways by other platforms. So we use this as a place to bring people in, but very little happens here that's active, because maintaining a website is quite a labor-intensive activity. We'd rather have a landing page with automatic aggregating ability rather than maintaining something explicitly. So that's a bunch of the data sources. In terms of this meeting today, then contributions to the things we're discussing can be given in the questions and chat window, obviously, of this platform. Any of the things I just mentioned can be used as a way of conveying suggestions to us. There's also a very busy Twitter feed that we use, so it's if you're a Twitter user, it's very easy to get in touch with anyone on the project using that platform. Obviously, the suggestions have to be fairly short. And at the last resort, there is an email address. We just discourage email because, of course, it's bilateral, so it becomes a private conversation. It's very easy to default to that rather than have a conversation in public. But if you can't find a way of communicating with the project quickly, then that's absolutely fine to use email. Whatever works for you will work for the project, provided that you make it clear that those contributions can go in the public domain. Okay, I think that's all I wanted to say about the data sources. I guess maybe if I pass over to you now, Mark, you can give an example of this in the way in which you use Data Warrior to interrogate some of the data in the project.
Yeah, great. Thanks. Thanks very much, Matt. Let me get that screen up. Okay, excellent. So I wanted partly to illustrate a couple of SAR points, which or SAR analysis points, I suppose, which is perhaps a theme of a couple of these meetings previously that Actelion and Isabel in particular have taken us through, and partly just to to do that with this project as a vehicle, and maybe some of you might feel inspired to do similar analyses or even use some of these methods, and give us your thoughts as a result of those. So hopefully everybody can see this. So what I started with, so background, Data Warrior it's a free-to-use SAR analysis tool that Thomas Sander has put together, and that's a link to download it. So you'll get these slides afterwards, and you can follow the links and stuff. But if you just Google Data Warrior, you'll find it. And these slides have within them some fairly detailed notes on some of the slides, because I haven't gone through in great detail how to do stuff on the slides themselves, because I don't want to take the time over that today. But the information on exactly how to do how to generate each of the views that I'm going to show is in the notes on the slides themselves. So in addition, there's a text guide to Data Warrior, which is a link up there, and a 14-minute video that my colleague Caroline Lowe put both of those together. So in terms of this project, as Matt has said, you can get the data from several places. I started with Google Docs last week and downloaded it as an Excel. And then actually to put it into Data Warrior, I needed to do a couple of things. So the EC50 mean column, which is an aggregation of all of the different falciparum dose response values that have been tested, is very useful, because obviously they've been tested. Some of these compounds have been tested in lots of different places, but it did actually, when I downloaded it, have this character string in some of the rows, which I needed to get rid of so that Data Warrior would treat the column as numeric, and unfortunately that was also present in the SD file when I downloaded it. So I deleted those divide by zeros, and I filtered to series 4 that Matt's been touching on. I also deleted the Inchi and Inchi key, because Data Warrior got confused about what's a column there. I think it was to do with probably commas in there. So I put a link to the Excel for series 4, having processed as above, though that will obviously be a static link, so better to go back to the sources that Matt mentioned about, particularly as time passes. So then I pasted those, just copy pasted those rows into Data Warrior, and there's so that's available as well. This is roughly what that looks like, and that's a link to that is in the notes of the slides. So then one thing that people often want to do in looking at SAR is think about, well, is my potency or other properties, are they driven by lipophilicity? So this is just a simple plot of lipophilicity, and this is C log P as calculated by Data Warrior against the log of the mean activity in micromole on the y-axis. So I've also colored the points by experimental log D. These points were generated by Monash, and you can see the values there that I've labeled them up with. They are pretty much in accord with the C log P, more or less, which is good. You can also see that there aren't many compounds with a lipophilicity below 2 which are potent. I guess there's an outlier here at sort of 1.5 at about 100 nanomolar. But nevertheless, I think potency in this at this sort of level in the sub 3 lipophilicity range is not bad. I've certainly worked on projects where you've had to go a lot more lipophilic than that to get reasonable potency. In terms of just extending that a little bit, looking at some of the compounds that are on this lipophilic ligand deficiency edge, if you like, then you start to see that at a lipophilic ligand deficiency of 5 or thereabouts, that some of these compounds, and so just I'm not demonstrating how to generate these plots. You can do so for yourselves. The instructions are in the notes of these pages. So if I just scroll up, when you get the slide deck, you can go to the notes pages and have a look at that if you want to. So those are some of the more lipophilic ligand deficient compounds as measured by potency and C-log-P. The other thing I wanted to demonstrate is just Matt mentioned in some of the potency work matched molecular pairs. So this is something that you can do. At least it takes a little bit of fiddling about in Data Warrior, but this is the kind of plot you can get from doing that. So here I've focused on compounds where there are pairs of compounds that differ by just a small change. In this case specifically, they differ by a variation in the scaffold. Okay, so this series is characterized by 5,6 core, and these are 5 of those cores. And so what you can see, for example, in looking at the pink and the red lines, so the red line links two compounds that differ by the presence or absence of a nitrogen in this 6-membered ring. So there's an extra nitrogen in this one, perhaps slightly obscured by the blue line, but you can see the core here. So it's pyrazine core, whereas this one is pyridine core. And so the pink line is a different set of decoration of that 5,6 system, but it's the same cores. So you can see that, you know, the extra nitrogen seems to provide some additional potency, which of course is good news because it's also providing some additional polarity. So this is buying you potency and polarity, which is always a nice combination to have. This trio of compounds shows something about variation in the 5-membered ring, and nitrogens there. So there's the 2 nitrogens in the triazole ring here, and then there's 2 flavors of pyrazole here. And then finally this one here shows that this additional nitrogen seems to be that there's some conflicting data on this in terms of whether or not that's good for potency or not. So if you compare these 2 compounds, then it's definitely bad for potency. And if you compare the 2 pale blue compounds, it looks to be good for potency. So maybe, you know, these are 4 compounds, perhaps they weren't all tested in the same assay. I haven't checked that. Maybe this is grounds to retest something like that. So anyway, this is a matched molecular pair analysis. I should say, in doing that in Data Warrior, you need to put in a core, and this is the core as it was defined. Actually, if you want to repeat this yourselves, you can either pick up the version of Data Warrior that I've got where I've already done it, or there's some macros that are recorded so that you can replay those macros. And in fact, some of those macros will help you do some of these steps even on different compounds with different cores, etc. The macros could still be useful. My final slide is just to say that obviously, as well as doing a matched molecular pairs on analysis on a core, you can do it on other substituents, and this is just an example on R3. So again, if you follow through one of the lines, you get to compare the impact of R3 on you know, different substituents on R3 and their potency. Clearly you can do that for metabolic stability where you've got enough measures, or you could do it, you could have lipophilic ligand deficiency on the y-axis, which is something I quite like to look at. Obviously, if you do want to look at lipophilic ligand deficiency, you need to be aware that quite often the ones that look quite good will actually be really polar compounds that are completely inactive. So you probably want to highlight those data points that are inactive. So Matt has included a qualifier for those compounds that are greater than some level of potency. So again, this slide deck will be available to you, and all the notes on generating the different views are in the notes part of the slides. So Matt, I'll hand back to you for the scientific questions.
Great, thank you. Thank you for that. I guess that demonstration shows the value of playing with the data in terms of generating new insights on what should be done next. And I guess I can't emphasize that enough, that it's incredibly valuable to have that kind of analysis done by anybody. That, you know, with more eyeballs on the problem, then all bugs are shallow. You know, if people would like to play with the data and suggest compounds in that way, that would be absolutely priceless for the project. So just in terms of a few questions that we can do in a little bit more detail here, I'd like to talk about three things just for a few minutes each. So solubility and this in vitro in vivo correlation issue, and then potency. Those are the three main things that we're looking at in the project. And the links are shown on the screen here. So the GitHub contains all the relevant issues, and if you just swap out this X for this number here, you'll get to the relevant page, which contains all of the relevant background information and links to other places where there's data. If there's time at the end, I'll talk about the other three discussion points, which are interesting in other ways. And I think what I'll do is I'll just run through a couple of slides on each, and then maybe pause after each to see if anyone has any immediate questions or comments. But then we can have a discussion time after I've been through a few of these things at the end. So solubility, I guess, is one of the main things that we have always had as an issue on this project. This is series 4, and all the series that we've investigated to date, we've always been very pleased with making extremely potent compounds, and then sadly realize that they are very insoluble. And this is a typical medchem problem, I guess. And series 4 is typically not really any different from that. We can regularly generate compounds that have nanomolar potency, but at the expense of problems with solubility. Chris Swain has done a nice analysis on many of the compounds in the series to show that predictions by software, such as ChemAxon, gives a good approximation to how soluble the compound is. So we can kind of believe some of the predictions that we're making in terms of solubility based on C-log p-values. And in many cases, we have been making molecules with C-log p-values that are far too high, and we've informally in this project said that we should be making new analogs that have calculated log p-values, C-log p-values of no more than about 3 or 3.5, just as a ballpark figure. Now there are exceptions to this. There are a number of compounds which are reasonably polar and demonstrated good solubility values with good associated potencies, and Mark just presented some of those. We did have this one collected data set of 8 compounds that were analyzed at the same time at the Monash Institute of Pharmaceutical Sciences, in which the solubility was measured along with the microsomal clearance, and the potency values in micromolar are given in red here, along with the solubilities that were measured. Now I think that these compounds at the time were chosen to be on the sort of better end of the spectrum. So these are the ones that I think were just featured as the specific data points in Mark's analysis. And there are some good compounds here. So particularly in the bottom right, we have this compound, MMV 67944, which has good measured solubility values and a potency of 140 nanomolar. The clearance times are a little too fast, the half-life is a little too short, but that's not bad. So there are some exceptions here to the rule, and some of the rest of these compounds obviously have nice polar groups on which are generating decent solubility values. But I guess these are some of the outliers, and in many cases we've been making compounds that suffer potency, a drop in potency when we make them more polar. Now, some independent analysis by Matt Tarnowski, who used to be here at Sydney and has since left and works at AZ, did the same kind of analysis that Mark was just talking about, and showed that that we do tend to get improved metabolic clearance values with improved solubility. So here with these 8 compounds that were measured at Monash we also got log D values, and sure enough, you improve the solubility and you improve the half-life. So generally speaking, I think we do want to push down the C log P and the log D to sort of improve these values. But I guess there's at the same time as assuming all of this, that we need to improve these values, generally speaking, we've also been looking at some of the SAR data and looking at all of the compounds for which we have clearance data, and these are all pictured on the wiki. So I don't have the space on the screen to look at them all. But if you go to the wiki, there's one big panel where all of the metabolic data that we have is shown. And if you take a step back from some of the data there, you can see that some of the strategies that you might adopt to reduce the clearance values, the metabolic clearance values, don't really tend to be having a lot of impact on those metabolic clearance values. So the standard tricks that you might have of putting blocking groups in benzylic positions and other tricks aren't really having a huge impact on improving the clearance values. So some in silico analysis was done, shown on this slide by Chris Swain, to show the sort of metabolic hotspots of some of these molecules. We've had some metabolic ID work done on these three compounds, and the result of that is shown very schematically in the bottom of this slide, which indicates that some part of the molecule in green, the circle in green there, is mono-oxygenated, and it's likely we're seeing dealkylation, where the part in green still has the oxygen attached, but the rest of the compound is falling off. But we don't know where within the green area this molecule is being oxygenated. So we don't really have a lot of sense of which part of the molecule is the problem. Nitrogen-based heterocycle like this often suffer from clearance by aldehyde oxidase, and Scott Oback at Pfizer very generously did a screen of a bunch of molecules, series 4 molecules, in his assay for AO activity, and the results are shown on the wiki again. But essentially, there were some molecules that were substrates for AO, but there were lots of molecules that weren't, and the picture wasn't terribly clear from that, other than to say that the amides that I'll show you in a minute, so these are the ethers here, the amides in series 4 were slightly more susceptible to clearance in that assay, and so they were slightly deprioritized in this project. But I guess one of the theories that then cropped up when we last discussed this was that, well, maybe we should be dropping the idea of blocking things to improve metabolic clearance, and instead just trying to improve the solubility as a gross parameter. And that if we improve the solubility well enough, then we will also improve the metabolic clearance. That was the idea. So ultimately, I guess the questions come down to, is it the expectation of people that is true, that if we improve the gross solubility of the molecule, that we will also improve its metabolic parameters, and that if that is the case, if that's agreed to be the case, then what strategies should we pursue to increase the solubility, which perhaps we haven't addressed yet? We've tried some obvious things of replacing aromatic rings with pyridines and supplying the molecule with hydroxyls or amines in various positions. But I'm sure that we haven't thought of everything, and I'd be very grateful and interested for suggestions as to maybe groups that we can include which might improve solubility. I guess that's essentially our main challenge at the moment. Now, I'm happy to take questions or suggestions now very briefly. But I guess I would, if anybody would like to say something very briefly, I'm just conscious that we only have an hour and we have to take questions pretty quickly. Otherwise, I can just specifically to do with this issue of solubility. Otherwise, we can move on to the next.
There's a couple of questions. Matt, shall I unmute people? So, Chris,
Cool.
did you want to? Chris Southern, did you want to ask a question?
Am I live?
Yep.
Yeah.
Yeah, no, I just said it's interesting to see the metabolites, whether the activity is dropping off as they're formed, or some of the metabolites are still active. We don't have to. You could synthesize them or just incubate them to see when they're generating and whether there's any activity in those samples.
Yeah, I guess we haven't done that because we're not completely sure. Well, so we haven't synthesized any compounds that are suspected metabolites, and isolating them from the assay with something we haven't done yet. But it's perfectly possible we could try that. It's not a route that we've gone down.
Okay, great. And I think Anthony, did you want to say?
Oh, hi. Yes. Dr. Todd, I was wondering if you ever tried a right-hand pyridine. I found a chloropyridine aldehyde from Sigma, and I know the Sidney Grammer unsubstituted ones were pretty bad, but a chlorinated right-hand pyridine or even a sulfur group like a sulfoxide could possibly improve solubility, at least in silico.
Okay, thanks. Yeah, I think the chlorinated pyridines would probably be fairly chemically reactive. That's a lovely group that we like to use in heterocyclic chemistry for substitutions in that chlorine right adjacent to a pyridine. But absolutely, you know, a substituted pyridine. I think there's a compound with a it's a CF3 group next to the pyridine in the northeast was perfectly potent and very effective. So I think that's something that we could is definitely a good strategy going forwards as a group that we know will be accepted there. Yeah. All right, look, given the time, I think maybe let's move on to the next thing. But please just keep those mental wheels turning for any possible groups that we could add in that are polar and which might be possible as the next thing we could try.
And also, I just wanted to chip in. Please keep contributions coming in through the questions with question section as well. We will get all of those recorded afterwards. Thanks.
Yeah. So the second issue was about this IVIVC. So we have three molecules in series 4 for which we have in vivo data as well as in vitro clearance values, and these are the three that are shown here. Now, the importance of this, of demonstrating whether there's an IVIVC, an in vitro, in vivo correlation, is so that you can believe the much faster to obtain and much cheaper to obtain in vitro data of microsomal clearance, so that you can say, well, the in vitro values are a good proxy for the in vivo values, so we should believe them. We only have 3 values, and here they are. And you know, if I would ever try and argue a trend from 3 data points, I would fail myself at my own undergrad exam. But you look at these 3 and you think, well, if you just look at the 2 right-hand compounds, so the compound in the middle, 565, and the compound on the right, and you look at the RAT, the RLM data, and then the in vivo clearance in blue, there's a kind of correlation. It's embarrassing even to argue that from 2 data points. So the kind of correlation that you think, well, maybe the in vitro data is looking okay, and we can believe it. But then you look at the left-hand compound, which is an amide, and which is a less potent compound. It's the red value of micromolar is the potency against the parasites, and that doesn't really match. So in order to continue to acquire in vitro data and to believe those data, I think it's important that we have a bit more certainty about whether we have this IVIVC. And so to do that, we would then need a few more data points. And I guess the question that I'd like to ask the community is which of the compounds that we have in vitro data for, or which compounds for which we have existing data, should we acquire the other data? So on this page, I've put 3 compounds for which we have some in vitro, and which are varied and which have certain features. So there's a couple of amides, there's an ether on the right, the compound on the left is potent and has a reasonable half-life. The one in the middle is okay potent, but has a worse half-life, and the compound on the right we know is an in vivo active. And it seems to me that if we could acquire another few data points, we could maybe have a bit more certainty about this correlation. So it's difficult to, just to go back to the slide, it's difficult to for people to assess the data within the meeting live like this, but I guess based on all of the structures that we see on the wiki, and I would direct people there now or after the meeting, if people had suggestions about which compounds we should send to Monash to be evaluated, then that would be very gratefully received. In case we're missing something, or in case it's obvious that we should be looking at some other compound, that would be a very, very useful input. Obviously, this is quite expensive work, so we want to make sure that we're choosing the right compounds. And I think in these cases, we'd have to resynthesize these compounds, so we've got to be sure. Very quickly, when we were discussing this with David Shackelford at the Monash Institute of Pharmaceutical Sciences, who's done a lot of, provided a lot of inputs on this part of the project. I'm very grateful to him for that. He suggested that one thing which we have no information on at the moment is this thing to do with volume of distribution. So how well a compound is distributed in vivo, and we don't have that, and yet this impacts half-life quite considerably. This is not something that I know much about. David was recommending we acquire this data, and for the two compounds that are in vivo actives, the ones that are shown here, he was suggesting that maybe we should acquire that data. I would be very grateful for anyone who has experience in that area to recommend yes or no as to whether we should be acquiring the data, that kind of data, at this stage of the project. How desirable that data would be based on what we know of these compounds so far. So the specific questions I guess I'm asking is, do we have an IVIVC? I'd suggest that we don't, because we have 2 or 3 data points. If not, which compounds should be used to acquire more data, and is there enthusiasm for securing VOD data, which again is expensive, so we have to be clear that we need it. So please feel free to suggest now or after the meeting. Very interesting suggestions coming in the question box, which I'm going to have to find time to look at here. We've got just 60 minutes left, so I guess we can take those comments and peruse them after the meeting, which is very, very useful comments coming from people. Thank you. I'll quickly move on to potency, which is conceptually a little easier and quicker, I think, to describe, if I may. So obviously the 2 issues we just discussed are the most important for us. Like I said, we have regularly in this project generated nanomolar potent compounds, but with undesirable metabolic properties. The summary of the SAR that we've got today is here. It's a busy slide, but it summarizes a lot of the stuff that's shown on the wiki. This is the ether series, this is the amide series. Ethers have been prioritized a little bit based on the aldehyde oxidase data that we had. And here is a very brief summary of a lot of compounds that were tested, both within OSM and previous. We have seen quite striking sensitivity, potency sensitivity to small changes in structure. Some examples are shown here. Triazolopyrazine compound, this exact compound was potent, whereas the triazolopyridine compound, which was made by a student in Edinburgh University, was found to be not potent. And this series of values has been confirmed. We inherited a couple of these values, and we've since confirmed them again, because we couldn't quite believe it, that a free amine here was potent. The dimethylamine was potent, but the monomethylamine was not. So there's clearly important groups in these positions that we have to worry about. And also in the northeast, very small changes to substitutions have had big impacts on potency. It looks as though an aromatic ring with something in the para position is necessary, but we can't be certain of that yet. So again, this is, I guess, food for thought here about what maybe we can try next. And if you browse the wiki, I think it's quite easy to see things that maybe will help you come up with possible suggestions for next compounds to make. Just to flag up that at the moment, right now, these compounds are under evaluation at Syngene in India. A set of compounds that were suggested by Chris Sway up here with a couple of chlorines on this ring and a blocking group. This middle set of compounds were made by a group of undergrads at Sydney University as part of a lab class. Ed in the lab here, Ed Sir, has been making thio series, so the thioether and the sulfoxide and the sulfone, just to see if that's an acceptable replacement for the linker, as well as this triazole. Obviously, click-based strategy is easy to make, and Alice has made this this alcohol compound, obviously as an attempt to make a polar compound with potency. So these are under evaluation. We were hoping to get the data back by today, but there wasn't quite enough time. And the ones that have been making at the moment, Ed is looking at the homologous thio series here. Chase Smith in the US and Massachusetts is making the reversed amide, which is going to be an interesting one to evaluate. Ed's also making the ketone here. Alice is on the dimethylamine here to check that value. The homologous amine, obviously, for obvious reasons. Ed is also looking at this compound here. This compound was evaluated without this methyl here, and was found to have reasonable potency, but terrible metabolic clearance. So we're interested in whether that methyl would block that. That was a suggestion by an undergrad who had a work placement in our lab called Tianyi. And then we are attempting to replace phenols with cubanes based on some work by Craig Williams in Queensland, and he's very kindly donated a couple of these cubane materials that we're now linking to the core to see if we can replace phenols with cubanes, potentially with improvements in solubility, but we shall see. As there are lots of possibilities for what to make next, and of course it depends a little bit on the data that comes in. Here are 5 suggested compounds, and there are others which we can discuss in the follow-up meeting in a couple of weeks' time. But here are 5 which we can use perhaps for a lightning poll. Is that something which is simple to do, Mark?
I hope so. I am just trying. It has been before.
So the compound on the left there is one that was suggested by Chris Burns at Wehi, this amide isosteer. The urea compound is perhaps an obvious change to happen here, which is a comment that we could make. This compound with something here, I'm not sure what this can be yet, but it was suggested as a possibility by a young scientist forum group at AstraZeneca, which Matt Tarnasky was leading. This is a compound that Alice has been looking at with very nice C-log P values, but has so far resisted synthesis, and I guess we're wondering if it's worth pursuing this compound. It's a nice one, but whether or not we should do that. And Chase Smith has suggested a scaffold hop to this kind of compound. So here are 5 possibilities that we're interested in, and I guess I'd be very interested to know whether or not people had immediate thoughts on which looked particularly valuable or of interest. So please feel free to submit a vote for this little poll, and we'll see what we get.
What I'm hoping is that I'm not obscuring the structures by getting the poll out there, but
no,
40%, that's okay, is it? So 40% of people have voted. So it's a bit of fun. If we could maybe get it to 70%, that'd be fab.
Chase is on the meeting, and he's in the lead here, so he must be pretty excited.
Has he been voting for himself lots of times?
He can't do multiple clicks. All right.
Okay, another 10 seconds. We'll close the poll.
All right.
So I think if I close the poll, all done. We're up to 64%. Anybody else got an opinion? Okay, so I think I can share that.
Well done, Chase. All right.
Excellent. So I think
obviously, Chase, you now have to make that. So, downside of winning is you're going to make it. Great. All right. Thank you very much for that, everybody. If anybody has any particularly strong feelings about these molecules and had a vote but has a backup reason for one being particularly good, then please do communicate that to us, because I'd love to know if there's one reason for we should be going for these rather than another. Okay, excellent. Thank you. Just very quickly to highlight a few other things. This deck will be available after the meeting for download, so you can peruse the diagrams. But just to highlight a couple of things on the last 3 discussion points, we suspect the mechanism of action of these compounds is binding to the ion pump PF-ATP4, because Kieran Kirk at ANU has screened a bunch of these compounds and found in his assay, which is an ion regulation assay, not a binding to PFAT4 assay, it's an ion regulation assay, that compounds that are active in his assay are potent. So the activity in his assay correlates well with potency versus the parasites. There appears to be a causative relationship. However, there is no crystal structure of ATP4. It's a big membrane protein. No one's crystallized it yet. There is a homology model. But one of the interesting things about this target is that it is the suspected target of a bunch of compounds that are in development, including the Novartis spiroindolones, and a bunch of other compounds that are in late stages of development. It's suspected to be our target. And when Kieran screened the malaria box, all of these chemotypes hit. Very bizarre and interesting that so many different chemotypes can apparently hit the same target. So the question is whether this is real, whether these are all in fact hitting the same target. If they are, how are they doing that? And so this is one of the features of the project that we're doing at the moment, is to try to develop a pharmacophore model for this, for what can hit this target. We tried about a year ago to build a pharmacophore model. Murray Robertson did some volunteers for this and built a preliminary pharmacophore model, which was not predictive and didn't take into account stereochemistry of the compounds that were screened and didn't take into account the possibility of overlapping binding sites. So in the next year or so, we are going to be attempting to build one of these models to try to understand which compounds hit ATP4, whether they do hit ATP4, and be a bit more predictive about compounds that might hit that target. This is obviously strategically important because MMV have no interest in developing a bunch of compounds that hit the same target. So this year, if you're interested in cheminformatics and you want to get involved in this part of the project, that will be fantastic, because I suspect there's going to be a bunch of in silico analysis that needs to be done. Penultimate thing, sourcing compounds. In case it's not clear, the main roadblock we have is preparative synthetic chemistry. So synthesis, organic synthesis of compounds. We have had compounds contributed to the project from a bunch of different sources from around the world. Either people who've made individual compounds or lab classes, undergraduate lab classes that have made compounds. Alice Williamson at Sydney has really pioneered this here, including getting students at a local grammar school to make compounds. One of the features of the open source project is that you can federate it, so people anywhere can make whatever they wish, and it's particularly well suited, therefore, to crowdsourcing of compounds. If anyone here knows of a group of students who would like to get involved in real research, maybe as part of their lab classes, please say. I know Chase has been doing this. I think Robert Broadrup is on the call, who has also been doing this. It's a potentially very powerful way of making lots more molecules. We're exploring a link with Eli Lilly's OIDD group to see if they would be amenable to doing some synthesis for the project. And if anyone has any ideas of other sources of chemistry or compounds that we should be exploring, I would love to know, because that is our major roadblock. Last thing very quickly is interacting with the data. People coming to the project can sometimes come across blockages to interacting with the data well, because people can't find stuff. If that is the case, and if people have difficulty in interacting with what we're doing, we'd love to know. The important part of this thing about open source is you can change it on the fly. So if you have issues with getting to grips with data in the project, please say. You won't offend anybody, please say, and we can try and fix those things. It's important that we reduce barriers to participation as much as possible. So the last thing here is just to emphasize the way in which you can interact with this project and the way in which you can take part and do your own thing. In closing, I'd just like to thank Mark for setting this up. Thanks to Paulus and Chris Swain for contributing to setting this thing up. And obviously to all of the many contributors to open source malaria for their continued input to the science, without which none of this would be possible. Thank you, Mark.
Fantastic. Thanks very much, Matt. So does anybody have any last questions? Please raise your hand if you do, and feel free to make further contributions on the questions electronically as well.
I can give control back to you if you want, Mark.
I don't think I have a lot else to present. I was just going to say that you will all get links to all this information, a recording of today's session, and the slides. And of course, they contain links to all of the information that Matt's been describing, GitHub and all of the wiki and all of that. Obviously, at the end of the day, what we really are interested in is if you have some ideas for the project, comments on some of the 6 questions that Matt's raised. We would love to hear from you, and I think there will be another meeting in June. We're just trying to line up exactly what that's going to be. Sorry, there's one on this project on June the 8th, which Matt is going to organize. Excellent. Do you want to say anything about that, Matt? So you've already got a date and time.
Sure. So it's important that we collate any community inputs and contributions from this meeting, and we can discuss those, I guess, in more depth in a more open-ended setting. So in a couple of weeks, on Wednesday, 8th of June, at the same time of day, we'll have another online get-together where people can suggest things. The link is there. It's on a different platform, but you just need to click that link and you can go straight in and we can discuss things. Obviously, in the meantime, please, if you want to get in touch and suggest things outside of that meeting, before that meeting happens, then please just do so, and I'll collect together an agenda of what we're going to talk about. Again, on GitHub is this issue number 394, and we'll put together things we're going to discuss, and people will be free to ask questions in a slightly more open-ended way.
Fantastic. Thank you, Matt. And then I'm also trying to line up a meeting for the 3rd Thursday in June, and we'd like to make that one a focus on the use of predictive models. So if you have some thoughts on that, or you have some experience of that, it would be fantastic if you were able to make a contribution. It doesn't have to be for the whole session by any means. It could be 10 minutes if you want. And in July, I think our focus is going to be on Kemble. So thank you very much to Matt for putting all this together, and also keeping us so nicely to time. Fabulous. Thank you very much, everybody, and I wish you all a good day.
Thank you for coming, everybody.